everyone for coming today to listen to this presentation. So it's a pleasure to be here, and today I'm going to present some of our work at Synlogic, and I hope that this presentation can illustrate what we're trying to achieve. As you know, here, and I've seen some members of other companies, microbiome companies, the microbiome has really gained a lot of interest in the recent years, and there's now a number of companies that are developing microbiome-based therapeutics. So I listed a few companies. If your company is not listed there, I'm sorry, I couldn't list all of them, but those companies are in a lot of various disease areas, and I'm just going to list like a couple. Infectious disease is a big one. There's a big push to treat Clostridium difficile using microbiome-based strategies. There's also companies that are working in immunology and inflammation, and those companies are using different modalities. There is the approach of the microbiome is very complex. It's maybe too complex, and we're just going to work on fecal microbial transplant, where the entire microbiome is transferred to a patient. There's other companies that use defined consortium of bacteria, so that's what Series Health is developing. Other companies are more focused on one probiotic strain, one specific species. We at Synlogic, we have a different approach where we take a very well-defined probiotic, a very well-characterized, and we are engineering it to add a function to the microbiome it's normally not there. So this is our vision at Synlogic. So the two pillars that we are based on is on one side synthetic biology, where we are engineering a bacteria using the tools of synthetic biology, and we engineer that bacteria to either degrade a toxic metabolite that accumulate in a disease state, and I will give examples of that. And then the other way of approaching it is by secreting an effector that has some activity, beneficial activity. And the other side of the company is like the biotic side, is like we're using a strain, a probiotic strain of E. coli called E. coli Nissl. And there's multiple reasons why we use that strain. Number one, it's been used for many years in Europe, especially. It's commercialized under the name Mutaflor. It's been isolated from the gut of a German soldier during the First World War, and that soldier was resistant to dysentery. So I don't think it's known if Nissl was actually the cause of the resistance to the disease, but nevertheless, it was isolated then, and then it's been demonstrated to have a lot of beneficial properties in terms of protection against pathogen and just in general gut homeostasis. And the good thing about this probiotic is that it's an E. coli, so we can really use almost all the tools in synthetic biology to engineer that strain. So the idea is that by acting from the gut, engineered bacteria can have a systemic effect on the rest of the body. So we hope that I hope that I can convince you today that this is the case. And feel free to interrupt if you have any questions. This is really informal, so if you have any questions, don't hesitate. So this is our current pipeline at Synlogic. There's two major areas. One is Inborn area of metabolism. The two that I'm going to talk about today are disorders related to hyperammonemia, where ammonia accumulates to toxic level systemically. The other one is phenylketonuria, where the amino acid phenylalanine accumulates to toxic level in those patients. But we have a number of other projects, especially in the immunology and immuno-oncology. And but I won't talk about that today. But there's also great progress that we're making in those areas. So here's like an overview of our current drug called SynB1020. It's a bacteria that's engineered to convert ammonia to the amino acid arginine. And by giving this what we call synthetic biotic orally, I will show you that you can actually lower systemic ammonia from the gut. So this, the patient population that we target, there's two of them. One is the urea cycle disorders. Those are patients that have mutation in any of the enzymes in the urea cycle, which is the cycle that converts ammonia to urea. And those patients are, the standard of care is like a strict dietary restriction in terms of proteins. And they also receive ammonia scavengers like Ravicti, which is a compound that can scavenge ammonia and gets excreted into urine. The other population of patients is not a genetic disorder, but it's acquired hyperammonemia called hepatic encephalopathy, or HE, where patients have liver damage that impair their urea cycle function, and so they start to accumulate ammonia as well. So the goal is to normalize systemic ammonia level with SYNB1020. So I talked about that already, but just a few points that we think that there's an opportunity there because, especially in urea cycle disorders, because we think that Ravicti is a treatment that can be improved, and we can also add to that treatment potentially. And hepatic encephalopathy is, we think that we also can access this disease because in this case, ammonia is produced from the gut by the microbiota through the action of ureases that converts urea to ammonia. So we think that for HE, we are really at the site of action with our drug. So here's a little bit about where does ammonia come from in the gut. So here's the large intestine, and ammonia in the large intestine comes from two sources. One is urea, the other one is dietary or endogenous proteins. And the microbiome degrades those compounds to ammonia by the action of ureases or proteases. Now this ammonia gets absorbed, gets into the circulation, and most of it will be converted to urea. But part of that urea will recycle through the gut, but the majority will be excreted in urine. So there's this constant cycling happening in our gut where ammonia just cycles between the systemic circulation and the gut. The other source of ammonia is through the action of intestinal glutaminase that are in the intestinal epithelium, and this will have systemic ammonia. So we think that with our treatment, we can capture this source of ammonia. This is a schematic of the current strain, SYNB1020. So there's a few characteristics of that strain. So it's an E. coli Nistel strain. And it's also a strain that's where all the constructs have been integrated into the chromosome. There's no plasmid. So to avoid horizontal gene transfer. And so there's the idea is to convert ammonia here all the way to arginine. Arginine, which is a beneficial compound for UCD patients especially. So to do that, we deleted a repressor of the arginine pathway. That's the first thing we did. Then we also expressing a feedback resistant RJ, which is the enzyme catalyzing the first committed step in the pathway. Then for biocontainment purposes, we also deleted an essential gene called Thia. It's a gene that is important for thymidine production. So this strain won't be able to replicate in vivo and outside the host. So we're not trying to colonize the gut. It's not, at this stage, we're really trying to have a therapy that is not like a bacteria that will be long-lasting in the gut. And that's mostly for safety measures. And then another modification that we're doing to the strain is that we are evolving the strain to become rifaximin resistant, because rifaximin is used for covert HE. So it's an antibiotic that's believed to act by decreasing the amount of microbes in the gut, so there's less ammonia production. So we want to have a bacteria that's compatible with that treatment. So how does the strain perform in vitro and in vivo? So we developed an assay to measure ammonia consumption in vitro, and you can see that CNB-1020 is able to hyperconsume ammonia in that. This is a microplate assay. And concomitantly, you see a production of arginine in the clinical candidates. So we decided to test that strain in an in vivo model of hyperammonemia. There's this model called the SPFH mouse model. It's an OTC deficiency, so the mouse is impaired for the conversion of carbamyl phosphate to citrulline. In this mouse model, you can really artificially increase their systemic ammonia level by placing the animals on a high protein diet. So the animals get extremely sick, and if you leave the animal on that diet long enough, it's lethal. But if you, so here you can see here the control ammonia levels are slightly elevated, but pretty normal. But once you place them on a protein challenge, then you get to like a really high level of ammonia. Now, if you dose those animals with a control strain, a Nisol delta Phi strain, you do see an effect already of ammonia lowering, so which suggests that Nisol is already something. It's not nothing. It's like Nisol has an activity in itself, inherent activity. And if you now engineer Nisol to produce arginine from ammonia, you lower the systemic ammonia even more to almost normal levels. But what was really interesting is that this lowering in ammonia is associated with an increased survival in an acute model of hyperammonemia, which is shown here. It's like you can see that the animals that were treated with CNB-1010 live like 2 days or 3 days longer, which might not seems impressive, but in the case of SPFH, when the animal gets extremely sick, that's actually very significant. So we were very encouraged by those data, and then so before I move on to the phenylketonuria project, this bacteria was moved on to phase 1 clinical trial that was just completed this year, and it was shown that this strain in healthy an individual was very safe and very tolerable, and so we're able to determine that this is a viable drug to get into phase 1B and phase 2. So I'm not going to talk about that today, but just want to mention that to you. The second project I'm going to talk about is it's phenylketonuria. So phenylketonuria is a disease where a patient accumulates a toxic level of phenylalanine. So phenylalanine is an aromatic amino acid that's normally converted to tyrosine, but a mutation in the gene making that conversion leads to the buildup of phenylalanine. Similar to UCD patient, those patients are on protein restriction diet to limit the dietary intake of phenylalanine, and they also have standard care is for a certain type of phenylketonuria is Kuvan, which is a cofactor. And then right now there's a PECPAL in phase 3 for this disease. It's injectable. It's an enzyme that converts phenylalanine to transcinnamate, and it's promising, but it's pretty. You have to inject yourself like multiple times a day, and so we think that there's like really an opportunity to find a treatment that can work in combination with PECPAL, or maybe work better than PECPAL for those patients. So why do we think that we can access systemic levels of phenylalanine? And so I put this slide there just to give you a sort of the context of how we see how we could access that systemic level of phenylalanine. So free amino acids in the gut come from two sources. One is the dietary proteins that get degraded to oligopeptides or free amino acids. The other one is endogenous proteins. And actually, if you look in terms of amount of proteins, this is a larger amount than the dietary proteins. And this include secretions in the gut, so digestive enzymes, mucins, serum albumin, immunoglobulins, and epithelium proteins coming from the natural recycling of the epithelium. And so they get absorbed into the portal blood, and then through the liver, they get into circulatory system. And then they recycle through those secretory organs, go back in the gut. So there's this, again, similar to urea, this constant recycling of amino acid between the circulatory system and the gut. So we think that phenylalanine that's produced in the gut can be sequestered by our microbiome, by our therapy. So we confirmed that by injecting animals subcutaneously with labeled phenylalanine, and so this is in mice. And you can see that when you do that, so you have a spike of labeled phenylalanine in the blood, transient accumulation in the blood, but you rapidly also see appearance of that labeled phenylalanine in the small intestine and to a lesser extent in the large intestine. So demonstrating that that phenylalanine was able to recirculate in the gut. So here's a schematic of the strain, the PKU strain. There's two conversions that we are using in this strain. One is the conversion of phenylalanine to trans-cinnamate, and this is similar to what PECPAL is doing. It's a deamination, and trans-cinnamate actually is a great biomarker and gets metabolized by the liver to hippurate, and hippurate gets excreted in the urine. So we can measure, looking at hippurate in urine, we can measure the activity of our strain in vivo. The other reaction that our synthetic biotic is catalyzing is the conversion of phenylalanine to phenylpyruvate through the action of an amino acid deaminase. So that's this protein is on the surface of the bacteria. The other protein pal is intracellular, and to bring in the phenylalanine inside the cell, we express a transporter called Phe to really improve activity of the strain. And PAL and Phe are under the control of what we call an FNR promoter, and that promoter is actually induced in anaerobic condition. So in the presence of oxygen, the FNR protein is inactive. When there's no oxygen, FNR, which is a transcriptional regulator, is able to bind to the promoter and activate expression. So we use this switch to turn on the microbe in vivo in anaerobic condition. And I mentioned that trans-cinnamate is excreted as hippurate that we can measure. And similar to the thioxotrophy, this strain is engineered to not being able to survive in the host and outside the host by deleting this essential gene DAPA. So how does this strain perform in vitro? So you can see that this strain is very active in vitro. It can consume phenylalanine very rapidly, a large amount of phenylalanine. And so we decided to move forward and test this strain in an animal model of PKU. So we opted for this model of PKU called ENU2. And those animals, if you place them on normal chow, have very significantly elevated levels of phenylalanine. You get to 1.5, 2 millimolar of phenylalanine. So we can reduce the phenylalanine levels in those animals by placing them on a fee-free chow and supplement phenylalanine in the water to sort of normalize their levels. And the way we do this assay is that we subcutaneously inject those animals with phenylalanine, and then we dose them with the bacteria, and we look at can we lower the systemic levels of phenylalanine doing that. So this is what you see here in this graph. It's like when you dose them with a control strand Nisl, you compare it with syn PKU, you see a significant decrease in the systemic level of phenylalanine. So it means that from the gut we were able to act systemically. As I mentioned, transcinamide is a biomarker of strain activity, and hippurate also is a biomarker of strain activity. You can see here that if you gavage animals with transcinamide, you get a 1-to-1 ratio excretion of hippurate. And so if you look at hippurate in urine and do a dose response with the bacteria, you see that a nice dose-response relationship, which is characteristic of a good drug. So what we're trying to do is we're really trying to apply pharmacology to our strain. I think that's this way differentiate us a little bit from the other microbiome company, where we can actually look at the specific metabolite, and that's a direct measure of strain activity. So we also tested this strain in monkeys, non-human primates. First, we showed that the recirculation is also happening in monkeys. When you inject monkeys with labeled phenylalanine, you can detect it in the gut. But also, if you inject this labeled phenylalanine and then you dose the monkeys with a bacteria, you see the appearance of labeled hippurate in the urine. So that was really, really encouraging. And then what we did next is we really wanted to see, is our strain efficacious in monkeys? So we did an experiment where we treated the monkeys with we challenged them orally with deuterated phenylalanine. So first we can observe that you see only the appearance of hippurate, labeled hippurate, when you dose the monkey with the PKU strain. So there's specificity of the signal there. But more importantly is that when you dose the monkey with the PKU strain, you see a decrease in systemic level of that phenylalanine that you gavaged the monkeys. So it looks like the strain was able to capture a significant amount of that phenylalanine that you delivered orally. And so if you take the AUC of this plot, this is the data shown here. Now, this is just free phenylalanine. So it's not really what you're going to see in patients, where patients are eating food that is a complex mixture. It's a protein. So is the strain going to be able to capture phenylalanine from proteins versus free phenylalanine? And so this is what we tested in this experiment, where we gavage the monkeys with proteins. And we looked at first the level of hippurate that we see in urine. And you can see that with the PKU strain, you see a great elevation of hippurate, which is a sign of activity from our strain. And again, if you look now at systemic phenylalanine levels after this protein challenge, you see a significant decrease, if you calculate the AUC, in systemic phenylalanine levels. So I think this demonstrates that the strain is actually active in vivo and efficacious in vivo in a monkey model, which is closer to humans than a mouse model is. So, I think in summary and conclusion, I think that we demonstrated that acting from the gut, a synthetic biotic microbe can have a systemic effect. And I think when we started the company, it was not an obvious statement that because of bioavailability of substrate, we were not sure that this would work, but now we're confident that we can have a strong systemic effect from the gut, acting from the gut. In the case of the UCD strain, we just completed a phase 1 study with a SAD and a MAD, showing good safety and tolerability profile for that strain. We also saw a signal in an ammonia-related metabolite and an arginine-related metabolite. So we think that that's a demonstration that the strain is active in human as well. And for the PKU strain, that's a second lead program. So this strain will advance to clinical stage next year, and so we hope that we can reproduce the data we saw in monkeys. So with that, I'd like to thank you for listening, and then I'm happy to take any questions. Thank you.